Hello everybody, my name is Ahmad Hosseinizadeh from UW Milwaukee. I work in the group of UMass, and this is the paper I would like to talk about today. Visualizing a protein quake with time-resolved X-ray scattering at a free electron laser. And this was published by David Arnold from Lutze group at Uppsala University last year. And you can see the full address of the paper at the top of this page. So in fact, the main speaker was Fatemeh Jamali, but she's on vacation and enjoying the summer today. And then she made me in trouble since yesterday. Very slow. So, okay. As an abstract, this paper describes a method to measure ultra-fast protein structural changes using time-resolved wide-angle X-ray scattering at an X-ray. And the main outcomes of this paper are as following. An ultra-fast global conformational change was observed. They call it protein quake. And this change precedes the propagation of heat through the protein. So basically two main conclusions of this paper. First, heat propagation, and then conformational change. So what is protein quake? As you know, some proteins harvest both the energy and the information content of light, and this energy can be similar to the protein's folding energy. Now, the main question is that how can the energy of light be usefully harvested without compromising the integrity of the protein? The answer they found in this paper was, in fact, protein quake. So protein strain is released as the quake-like structural deformation that rapidly goes away from the protein. The question is that how to probe this protein quake? So the answer is that experimental tools with picosecond time resolutions are required, at least picosecond. The basic method they used was a well-known method, of course not for myself, time-resolved X-ray scattering, briefly TR-wax, which is a synchrotron-based method that measures the nature and timescales of the global structural changes within proteins. But problem with the synchrotron is that time resolution at these sources is limited to 100 picoseconds. Therefore, let's say larger resolutions are required, and this can be solved by XFEL. Okay, the target sample to study this protein quake is the photosynthesis reaction center of Blastochlorus viridis. So let's call it RCvir. Usually this biological name are very difficult to pronounce, like my family name. So let's just call it RCvir. The major components of this particle are 4 subunits, M, L, H, and C. And then the other stuff you can see in this picture are the cofactors of the protein, such as BPH, BCH, and so forth. And the blue lines just specify the membrane part of the protein. This is a well-known setup for everybody at XFEL CXI station of LCLS, of course. So the green jet is just a sample. I mean, here is just a. Oops. Okay. Do you have any connection with you?
You're good. I'm
Okay.
still okay.
Okay. So this is the jet of particles, proteins, and this is XFEL beam, and this is the laser pump to photoactivate the particles before hitting them by XFEL, and this is the detector. So how they do photoactivate the samples? Photoactivation of samples was done by 800 nanometers optical pulses of 500 femtosecond duration. and the pulse fluence was 5.3 millijoule per millimeter square. So as you see in this picture, this is the absorption spectra from the RCvir list. The red one is after exposure to the single 800 nanometer femtosecond laser pulse, and the black one is just before photo activation. So you can see. I'm sorry? Yeah. So you can see slight differences between both. What about data collection? The X-ray images were integrated in rings. So they, as far as I understood, they took 9,000 good diffraction patterns, and then they averaged it in rings. The 9,000 diffraction patterns for every time delay. So the different scattering amplitudes delta S were collected as a function of time delay delta T between the optical laser and XFEL pulses, and data collected in a domain of 0.1 to 2.5 inverse angstrom. And here delta S is in fact photoactivated minus dark. Dark, I think, is just non-photoactivated, and which is given by this Q, this formula. Theta is a half of the deflection angle of the scattered beam, and lambda is just the X-ray wavelength. As the technique for study this or analyze this data, they use spectral decomposition. So TR wax data decompose into the 4 major components by spectral decomposition, and this exploits differences in the rates of growth and decay of each component to extract basic spectra. So these are the amplitudes of those components, and you can see its amplitude versus time delay, and delta is itself in terms of Q. So the main task remains to, let's say, assign a meaning to every of these components. Okay, so the 4 components of the TR wax data are named as following. C1 is just ultra-fast component, which is an average over the first 3 time points after photoactivation. C2 is protein component. It displays some oscillatory features here in this area of the protein difference scattering. C3 and C4 are responsible for heating propagation from the protein. So in fact, C1 and C2 are related to the protein quake, and C3 and C4 just address the heat flow through the protein. So let's first study the heat flow in this protein, how to find this from the cofactors. The method they used was a molecular dynamics simulation, and from this simulation they found this graph. I mean, some results which are shown in this graph. These are basically the temperature of cofactors versus time from molecular dynamics. So, for instance, the blue and dark are BCH cofactors. The red one is P960. And if you magnify this part, you can observe these details here. And the yellow one is for protein, and for instance, the gray one is for the surrounding solvent, and so on. As you observe here, the cofactors heated up to a few thousand Kelvin, but they cooled up quickly. For instance, by half in 7 picoseconds. Yeah. Okay, let's continue with that. And relate this C1 to the results of molecular dynamics. In fact, to find a correlation between molecular dynamics simulation and the experiment itself. So first of all, if you compare this yellow peak here and yellow for protein, I mean, with the peak of C2, or let's say C3, yeah. C3 and yellow one here, they have a peak at T equals to 14 picoseconds. So then one can say that C3, in fact, it measures the heat flow of the protein. And if one compare this gray one shown here with C4, they also agree with each other. The conclusion from this comparison was that C3 and C4 measure the flow of heat through the protein and into the environment. And now the question is, what about C1 and C2? Basis spectral C1 and C2 in fact describe the protein's structural dynamics. The growth of C1 is very rapid, it just quickly grows and maybe hard to resolve. C2 has some oscillations here. You can see both of them here. This is C2 again, and you can clearly see some fluctuations in this area between 0.2 and 0.9 Q. These oscillations were not observed from synchrotron data, so the first time they found it from BioXFEL experiment. And C2, in fact, reveals an ultra-fast structural change in the protein. Let's talk more about this plot. So in half maximum at delta T equals 1.4 picoseconds, C2, the half maximum of C2 is just at delta T equals to 1.4 picoseconds after photoactivation, and it peaks at 7 picoseconds, which is and decays with half-life of 44 picoseconds. But anyway, I mean, the main conclusion was that this structural deformation propagated away from the epicenter of the protein quake before thermal diffusion. I mean, as you see, C1 and C2 responsible for these structural changes are before C3 and C4. So they are, let's say, in advance of this heat propagation. So what about protein quake or conformational changes? How they found it from this delta S data. So by comparing predictions recovered from photo-activated and reference molecular dynamics, they found, let's say, an interpretation for C1 and C2. So this comparison was done by a chi-square fit. Chi-square fit was done to C2 by considering just the motion of the cofactors and frozen the other parts of the protein, and chi-square fitting to C2 was done by considering the entire protein is moving. So overall, they found an ensemble of 720 best fits from this comparison, and let's say those 720 pairs, in fact, are shown in this graph, in two graphs. As you see, the results of chi-square fit from those 720 pairs are in a good agreement with the experimental data in black color. So briefly, I would like to say that this chi-square fit, in fact, was done to find the best pairs which can be fitted to the experimental data. And those pairs were, in fact, 720 in number. So the ensemble of these 720 pairs was taken, and in order to find the conformations, they found the internal distance between the carbon alpha atoms of these pairs. I mean, the carbon atoms, for instance, for photoactivated and non-photoactivated pairs, and a matrix of internal distance was generated, and then the average and the standard deviation of this matrix was computed, and to find the let's say recurring internal changes, they put a threshold to identify these let's say little movements. The threshold was that the magnitude of the average of this matrix was 90% of the standard deviation of that. And also, each selected carbon alpha atom had at least 20 internal distance changes above this threshold. So The conclusion was that almost all recurring internal distance changes selected by the above analysis, corresponding to an increase of the internal distance between carbon alpha atoms and the RCvir in this transmembrane domain. So in fact, the main conclusion was that all internal distances show that there is an increase between the internal distance of carbon alpha atoms, not any contraction. So here in this figure, maybe it's hard to recognize, but one can see this is the reference, and this is the, let's say, the selected carbon alpha atoms are in sphere, and this is the movement of the carbon atoms compared with the reference. So, for instance, here you can see a little changes and so on. But in the movie, I will show in the next slide. You can see this in a better format. So the shift in the ensemble average amounts to a modest expansion, about 1 angstrom resolution, or in fact 1 angstrom amplitude of the protein. So in this movie, you can see if it works. Yeah. So there is a expansion, which is in fact increase the internal distance of the carbon alpha atoms. And okay, as conclusion, they found XFEL facilitates the direct visualization of ultra-fast protein motions using TR-wax technique, and the strain created by energy input to the protein cofactors is revealed via a structural deformation that propagates through the protein faster than the heat. Thank you.